Hello again. Now we're going to begin the topic of cancer. There are several short videos throughout this topic. Will point them out to you and ask you to please watch them. They'll help explain what we're talking about. They're mostly a minute to a minute and a half, so you should do all right with them. One of the first things I need to ask students about in this topic is if they know what certain screening tests are. My demographic knows all about these, but I have made the mistake in the past of assuming that you did. So as long as we go through this, I know that at least we're on a level playing field when it comes to discussing these screening tests. The first one is a mammography. Do you know what mammography is or what a mammogram is? Some of you might remember this when we talked about Angelina Jolie and her experience with having the BRCA mutation. A mammogram or mammography is a test where each breast is compressed horizontally and then obliquely or sideways in between this camera unit and a film plate, and it's not comfortable, but an x-ray is taken in each position. Radiologists then look at the results to look for abnormalities. This is a screening test for breast cancer. A PSA test is a screening test for prostate cancer. In case you've never paid attention to this, honestly, I'm not sure why you would, unless unfortunately you had a family member who's had prostate cancer. It's a gland in men that can become malignant. A digital exam of the prostate is done in the doctor's office. He's also eloquently pointing out, if you will, where the prostate gland is, but he's also examining it for changes to see if it's smooth, to see if there may be any abnormalities. That's one test for prostate cancer. But the PSA test, it's not a public service announcement. It stands for prostate-specific antigen. In the absence of prostate cancer, the cells in the prostate are healthy, they're organized in a tight pattern, and only a small amount of this antigen, this prostate-specific antigen, will leak out of the prostate gland itself and get into the bloodstream. However, with prostate cancer, cells become disorganized. We'll talk about that. The layers between the prostate and the blood vessels become disrupted, and more of this prostate-specific antigen can leak out into the blood as a result. A PSA test measures the amount of these antigens that are found in the bloodstream. Now that we have that kind of settled, let's talk about what's referred to as lead time bias. I'm going to talk about Beth and Nancy and their ill-fated trip to Washington, then explain how that also refers to cancer diagnosis, and opens up a whole lot of questions about screening tests. In this example, Beth is getting on a train in Boston. Nancy boards the train in New York. When they both get to Washington, the train crashes and both die. It may look like Beth was a longer-term survivor, but only because she got on earlier than Nancy. Now, how does that translate into cancer screening and cancer diagnosis? On the top, without screening, cancer starts. It could be any type of cancer, but one that takes many decades to develop. On the top example, the cancer diagnosed in this person was found because they began exhibiting symptoms at the age of 67. They were dead by the age of 70. The 5-year survival rate for this person was 0. They did not make it to 5 years once they were diagnosed. On the bottom, with screening, the person was diagnosed at age 60. This person also died at the age of 70. This makes the statistics for the 5-year survival rate at 100%, but they still died at the age of 70. The person on the bottom had 7 less years of being cancer-free because they had been diagnosed with a screening test. This leads to many questions about how effective screening tests can be. Do they prolong life? Or do they just prolong the amount of time that a person knows they are living with cancer? Because cancer is not one disease. Cancer is many different diseases, which we'll talk about. So screening tests for certain types of cancer is very controversial. Cancer is not a single disease. After you watch this short video, you'll better understand that there are hundreds of different types of cancers that can affect all the tissues and all the organs in the body. And there are many different causes of cancer, including genetic defects, viruses, the HPV virus, human papillomavirus. Strains of it have been shown to cause cervical cancer, chemical exposure, exposure to radiation, and sometimes it just happens. Sometimes there are spontaneous mutations that lead to the development of cancer. Apoptosis is another name for programmed cell death. Normally, cell growth is carefully regulated. Cells have a program within them that tells them when they're supposed to die. Red blood cells last about 3 months and are then replaced in the bone marrow by new red blood cells. The cells inside your mouth and your stomach are replaced every 3 days. So normally cell growth is carefully regulated. There are internal signals that tell cells when they're supposed to die, and there are external signals, the neighboring cells that send out signals to slow down the growth of cells. Apoptosis is programmed cell death. What happens during cancer is that cells ignore this program. They do not die, and they just continue to divide and divide and divide. There are what we call drivers of cancer. For example, proto-oncogenes. These are the type of genes that are involved in normal growth and division, but when they become altered, they become oncogenes. Proto-oncogenes are vulnerable to change, and when they become oncogenes, onco is a prefix for cancer, these genes then code for proteins that are mutated and can become cancerous. There are tumor suppressor genes that are also involved in controlling growth and division of cells. Cells with alterations in tumor suppressor genes may divide in an uncontrolled manner. And then there are DNA repair genes. We have genes that fix damaged DNA, but cells with mutations in these types of genes tend to develop mutations in other genes. And when the mutations that lead to cancer are not fixed, they're allowed to grow, and cancer can develop. So cancer is a disruption in the orderly growth of cells within the body. Internal regulation fails in that the proteins that normally suppress growth are no longer being produced. So these suppressor genes that normally curb growth stop working, and they allow cancer cells to form. For example, the BRCA gene is a tumor suppressor gene, and when it is mutated, cancer is then able to grow. External regulation fails. Transformed cells may no longer be affected by signals from neighboring cells that attempt to tell them to stop growing. They may no longer have the receptors that send out signals that tell cells to stop growing. They become what I call bad listeners. They don't listen to their neighboring cells, and they just keep growing and invade other neighborhoods, if you will. And they lead to the formation of malignant or cancerous tumors. A typical body contains about 100 trillion cells, and healthy cells divide to replace the cells that die. Carcinogenesis, genesis, creation of, CARCINO is a prefix for cancer. Carcinogenesis, formation of cancer. We have normal cells that may become abnormal cells by a process of initiation. This initiation can be caused by environmental agents, viruses, X-rays, ultraviolet light. These were mentioned in a previous slide, and they can form abnormal cells. We have many processes inside the body that can reverse or repair these mutations. But if they don't work, then there's what's called the promotion phase of carcinogenesis. More cell damage, more cell division, again because these cells are no longer listening to the apoptosis, to the program that tells them to die. The promotion phase can last for years. It can progress. This uncontrolled growth can continue to form the formation of tumors. If those tumors continue to grow, they may metastasize. Metastasis is what occurs when a cell breaks off from the original tumor from the site of origin, goes into the bloodstream or the lymphatic system, and spreads to other parts of the body. Now, metastasis is what occurs in what are called solid tumors, tumors of the brain or the breast or the lung or the prostate. Metastasis is not what occurs when someone has lymphoma or leukemia. These are blood cancers. Carcinogens are external cancer-causing agents. About half of Americans may develop cancer about 3/4 after the age of 55. And as I just said, metastasis is the transport and regrowth of cells from the primary tumor to other parts of the body. Angiogenesis is the creation of new blood vessels that provide the tumor, the cancer cells, with nutrients and remove waste products, enabling those cancer cells to continue to grow and create larger tumors and possibly metastasize. The risk of death by many cancers may be reduced by adopting behaviors, healthy behaviors. Physical activity, don't smoke, just don't. Eating as healthy as possible. Regular screenings may help reduce death. What I mentioned earlier, though, is that there are controversies concerning certain types of screenings. We'll talk about this in our discussion in class. Early detection and treatment may be critical to surviving certain types of cancer. It is important to know early signs and symptoms, and for some, self-exams and screenings. The ones I would recommend for your demographic would be skin cancer screenings, and I'll talk more about that when we talk about skin cancer. But I am curious to know what you think about the early detection and treatment after discussing lead time bias. Looking at some of the causes of cancer, we know that smoking greatly increases the risk of developing lung cancer. All right, cigarette smoking. There are a number of genetic factors that can play risk in cancer development. That was something many of you may have seen in your family tree. We also talked about the BRCA mutation, the mutation that suppresses the genes that create proteins that prevent cancers from growing. When that particular gene is mutated, the suppressing action no longer works and it allows cancer cells to grow. Alcohol has been shown to increase risk of a number of types of cancers, including liver cancer and esophageal cancer. Sun exposure, the ultraviolet from sun, can penetrate into the nucleus of the cell, affecting the DNA, enabling it to then lead to production of mutated proteins that create mutated tissue, resulting in tumors. And infectious agents, chemicals, can lead to development of cancer. One question I get a lot is, how can I change my diet to reduce my risk of cancer? Many of you see lots of information and misinformation in the media on foods that are superfoods, foods that you should eat, foods that you shouldn't eat. According to the National Cancer Institute, scientists have studied many foods and dietary components for the possible association with increasing or reducing cancer risk. But with few exceptions, studies of human populations have not yet shown definitively that any dietary component causes or protects against cancer. As much as we want to control our risk of developing cancer through diet, you need to know that there is no dietary component that has been shown definitively to protect or prevent cancer. Dysplasia or precancer changes in cells signal the presence of oncogenes, the genes that increase risk of changing structure of cells to increase cancer. Normal cells can become cancer cells. That's what we're talking about in this topic. But before the cancer cells form in body tissues, cells go through a number of abnormal changes called hyperplasia and dysplasia. In hyperplasia, there is an increase in the number of cells in an organ or tissue that appear normal under a microscope. But in dysplasia, the cells look abnormal under a microscope. They're not yet considered cancer, but they are signaling that cancer may be at a higher risk of developing. So these tissue changes may lead to cancerous tumors, or they may not. I know this is really confusing, but this is based on the information that we have. When we were first talking about the carcinogenesis or creation of tumors, at the beginning of that sequence of slides, you saw cells can change and lead to abnormal cells, but that because of various processes, those cells can revert back to normal. This can happen a number of steps along the way. But please understand that hyperplasia is more likely to revert to normal cells than dysplasia. This is something that you need to talk about with your doctor. For example, a woman may find with a pap smear, with the results of her pap smear, that cervical cells have become hyperplastic or developed hyperplasia. There's an increase in number of cells. This would be a reason to go back and have follow-up with your doctor, assuming you have insurance, health insurance, and support. Dysplasia, the doctor might want to have closer surveillance, or you may want to have the cells removed. Certainly, when cancer develops, the chance of reverting is very small, and you have to take measures. We have cancers in our lymphatic system. We talked about the lymph system when we looked at infection and immunity. Lymph is a clear fluid that carries those white blood cells. We talked about T cells and B cells being part of our immune response, and they congregate in lymph nodes. Lymph nodes are tissues that filter viruses, bacteria, and cancer cells. It's where those antigen-presenting cells meet up with the T cells and B cells, but they're also a place where cancer cells may metastasize to and spread throughout the body. And there are types of cancers that can develop in our lymphatic system.